Hello, Biochem class. Today I'm going to talk to you about something that I'm sure you haven't heard a lot about. We're going to be talking about coronavirus, but not just the regular coronavirus. Today we're going to be talking about a paper on the identification of the receptor binding domain of the spike glycoprotein of human betacoronavirus HKU1. This paper is by Zhao Hui Qian and others that I put in et al. All right, so we're going to start with a little bit of background information. To start, there are 4 different kinds of coronavirus. There's the alpha, the beta, the gamma, and the delta versions. The paper that we're going to be looking at focuses on the human beta coronavirus HKU virus, which was discovered in Hong Kong in 2004 and subsequently has been found in humans worldwide, where it accounts for about 0.9% of acute respiratory infections. So you may not have known it, but coronavirus has been around for a lot longer than people thought. Looking at this specific coronavirus, the coronavirus spike glycoproteins mediate receptor binding, membrane fusion, and virus entry and determine the host range. The large spikes on the envelope of the coronavirus virions consist of trimers of the 200 kDa spike glycoprotein that bind to a specific host receptor. They mediate virus entry, tissue tropism, and host range and can affect virus virulence. The spike protein is the target for coronavirus neutralizing antibodies and is an essential component of COVID vaccines and vaccine candidates. So this is important to us nowadays, as just recently it's been announced that the Pfizer vaccine is available for wide use. This is the kind of stuff that they're looking at for the one that is out now. So the purpose of this paper. In this study, they used the HKU1 protein-specific neutralization antibodies and N-terminal or C-terminal truncated S1 proteins to determine the location of the receptor binding domain in the HKU1 spike protein. In this study, they showed that for the spike protein of HKU1, the purified C domain downstream of the N-terminal domain could block HKU1 virus infection of human respiratory epithelial cells, and that several monoclonal antibodies that mapped to the C domain neutralized virus infectivity. Thus, the receptor binding domain of HKU1 spike glycoprotein is located in the C domain. All right, next we'll be looking at the materials and methods in this paper. Cells, lots of cells. So obviously, if you want to be testing a virus and looking for antibodies to use against the virus, you're going to need a lot of cells. So the cells that they primarily used were human tracheobronchial epithelial cells, or HTBE cells. These were used initially in the study to propagate the HKU1. The human tracheobronchial epithelial cells from donors without lung disease are cultivated to determine pro-inflammatory and antiviral responses. So these are used as a sort of baseline to figure out what inflammatory and antiviral responses can be found. All right, next is constructs. So the full-length codon-optimized genotype A of HKU1 spike gene preceded by a Kozak sequence was synthesized and cloned between the Hindi3 and the X-ball sites for eukaryotic expression. To eliminate a furin cleavage site and minimize cleavage between S1 and S2, both arginine 759 and arginine 760 were mutated with alanine substitutions in the HKU1 SAA construct. So that was done to remove the basic features of the arginine and replace it with an alanine that is much less basic. On the right, we see the series of deletion constructs that they used to, or that are used in the HKU1 that they amplified using the following primer pairs. I included this mainly just to show that there were a lot of primers, forward and reverse, that they used to specifically insert the portions of DNA that they wanted. And they inserted them between the NHEL and the BAMHI sites of PLG, which provides a CD5 signal peptide before NHEL and human FC tag after BAMHI. So this means it's going to be able to be detected much easier than it would have been if they had not included these. And it'll give a CD5 signal and an FC tag. So next, we'll be purifying the HKU1. So soluble S proteins were purified by affinity chromatography using either a bead column, which we've talked about before in class, for the HKU1 secto, or protein G column, which is a little bit different, but just the same concept, for an FC tag truncated proteins. And the purified S proteins were detected by Western blotting, which can be seen on the right. The blots were probed with horseradish peroxidase, and the concentration of the antibody was increased for each blot. So as you can see for the blots, which are in black and white, the more blots that they did for the same products, the less that you can tell that it's there. And the purity for each protein preparation was demonstrated by an STS page followed by a Kumasi staining. The Kumasi staining is shown with the gel that shows the blue lines in it. And it shows that they were able to separate it well and that the purity was acknowledged. So next up is generating antibodies. So of course, when you're studying a virus like this and you're studying the antibodies that go along with it, you need to make antibodies. So to make these antibodies, they immunized mice with 100 grams of the HKU1 sectoprotein at days 0, 14, 28, and 42. Then 3 days after that, the mice were euthanized and their spleens were harvested. And then the splenocytes within the spleens were fused with myeloma cells to generate hybridomas that were cloned. So these cloned hybridomas were where you get your antibodies from. So the antibodies to the HKU1 spike protein were detected by an enzyme-linked immunosorbent assay. So we'll be talking about this in a little bit, what the ELISA is, and also Western blotting and an IFA. Along with this, 2 rabbits were pre-screened as negative for the presence of cross-reactive antibodies to the HKU1 spike glycoprotein and were immunized with 100 grams of purified HKU1 sectoprotein at days 14, 42, and 56. 2 weeks following the final booster of the IgG was purified using a protein G column, which they also used earlier when looking for the purity. And the antibodies to the HKU1 spike protein were detected by ELISA, Western blotting, and the IFA, in the same way that they used it for the mice. Now, what is the ELISA? The enzyme-linked immunosorbent assay. So in this, I decided to put a little diagram of what this is, just in case you weren't aware of what the ELISA is. So the diagram is on the right, and then along with that, step 1 is to coat the ELISA plate with a capture antibody in excess unbound antibody, and then it's washed from the plate. So all the excess is removed, and what's left on the plate is just the capture antibody. Then the sample is added that you want to capture. Any antigen found on the sample will bind to the capture antibody, which in this case are looking for the antibodies for coronavirus that they got in the mice and the rabbits. Any excess is then washed from the plate. Then detection antibody is added. This antibody is labeled with an enzyme, usually the horseradish peroxidase. Detection antibody binds to any target antigen already bound to this plate. So it's binding onto the target antigen and the capture antibody. Then lastly, substrate is added to the plate. The ELISA assays are usually chromogenic, using a reaction that converts the substrate to a colored product so that you can see it without anything special, which can be measured using a plate reader. All right, then looking at the results of the paper. So first is the truncations. Truncations were performed to determine stability of folding in the HKU1 coronavirus. So this is a little bit like the Chen paper that we looked at last week on Monday. So we can see in figure A that they used different truncations to determine the length, or to determine a length of certain variations of the coronavirus. Both N and C terminal truncations were performed, with the N terminal truncation being the S295, the C terminal truncations S14 ones, and then both N and C terminal truncations at the 310. The S14 673 amino acid and the S310 673 amino acid proteins were considerably less stable, as shown in figure B, where you can see which are more stable by how prevalent the black line is for their lane. And they were considerably less stable than the other truncated spike proteins, so those could be ruled out as binding domains for the coronavirus. So by using this, and then using a Western blot, they were able to determine by prevalence of the black line shown which ones would be potential binding domains. All right, then looking at the C domain binding. Since the epitopes of the HKU1 virus neutralizing monoclonal antibodies map between amino acids 535 and 673, the people who ran this study reasoned that this most likely is a part of the receptor binding domain, which would make sense. Located within the C domain, which would be the S295 to 755 amino acid of the HKU1S protein. So looking at this, they performed some ELISAs again with the optical density reduced as more antibody was introduced for all the proteins. So looking at the figure on the right, it shows the optical density is the x-axis, or the y-axis, sorry. The y-axis is the optical density, and the x-axis is the antibody being introduced. So as you can see, as more antibody is introduced, the optical density is decreasing, and it showed that across the board, there was a decrease in all of the different strains that they used. Therefore, there was no difference in that respect. Now, testing their hypothesis. So S14 755 amino acid FC and S14 294 amino acid FC and S295 755 amino acid SC proteins were purified and pre-incubated with the HTBE cells to occupy the virus receptor on the cell surface. So they decided to use these ones because those seem to be the most prevalent and the most likely for the binding domain. So pre-incubation of the HTBE cells with the purified C domain FC at 3M markedly decreased the HKU1 virus infection and reduced virus release by 25-fold and 360-fold at 24 and 48 hours post-inoculation. The inhibition effect by the C domain protein was dose dependent. So the more dosage that they used, the more inhibition that they found. And they found that over time, it greatly reduced the virus release. Those results strongly suggest that the HKU1 RBD is located within the C domain of the HKU1 spike protein, likely between the amino acids 535 and 673. Going into the discussion now. So all the known Betacoronaviruses in group A normally use their NTDs to bind protein receptors or sugars. But surprisingly, they found that in the HKU1 virus, another Betacoronavirus in group A uses its C domain, not the NTD, to engage with its yet to be identified receptor, which at this point in 2004, they hadn't identified the receptor. I'm not sure if they have yet. Two Betacoronaviruses in group A, HKU1 and MHV, have evolved to use different regions of their spike glycoproteins to recognize the respective receptor proteins and trigger the conformational changes of S protein, leading to membrane fusion. Also, the neutralizing epitope is conformation dependent. So what that means for this paper in general is that the coronavirus groups evolved in different ways in microevolution, that their spike, or to use different regions for their spike glycoproteins, and that is big in helping to determine which antibodies to use, and also big in finding which vaccines can be used against different coronavirus strains. Also looking at the conformation dependency, that means if the conformation can be changed so that the C domain is opened up and purified for the HKU1, it will neutralize the HKU1 much better than previously detected. Finally in the discussion, So they identified the C domain as the locus of the epitopes recognized by two HKU1 neutralizing antibodies, and showed that the soluble C domain can induce receptor blockade, resulting in inhibition of the HKU1 infection. These data demonstrate that the RBD of the HKU1 is located within the C domain of the S protein, likely between amino acids 547 and 573. All right, and then in conclusion, these results highlight that several Betacoronaviruses in Group A have evolved to use different regions of their spike glycoproteins to recognize their respective receptors. These findings will be able to aid studies in the HKU1 virus pathogenesis and receptor discovery and facilitate development of new models for Betacoronavirus evolution. And then bibliography.